This is a production of Cornell University.
For our keynote speaker, Dr. David Wilt, and he did his bachelor's work at Illinois State University, then did his master's and PhD at Michigan State University, before going to the Baylor College of Medicine for a postdoctoral fellowship, where he studied reproductive physiology and endocrinology. After being at Baylor, he went to perform research at Texas A&M and the National Institutes of Health before moving to the Smithsonian Institution, National Zoological Park. Dr. Wilt is really one of the pioneers in taking reproductive biology, the science of reproductive biology, and applying it to wildlife conservation. He's worked with a wide number of species and has been involved really with really some of the most important and successful conservation efforts that have been achieved over the past decades, and he'll speak a bit about those today. His current position is Senior Scientist and Head of the Center for Species Survival at the Conservation and Research Center. He is in charge of the wildlife animal collection at the National Zoo's research and breeding facility at Front Royal, Virginia. And I know those activities with trying to manage small populations of endangered species for preserving the species and also for studying them and getting knowledge to help us preserve them. It takes a great deal of his time. As I mentioned, Dr. Wilt has really been involved in some of the most important studies related to reproductive physiology and its uses for conservation, ranging from the black-footed ferret, Florida panther, cheetah, but his work is not limited to reproduction alone. He's in charge of very holistic approaches to looking at the different problems that affect an individual species, and an excellent example of that is his work in China with the giant panda. He's recently actually edited a book called Giant Pandas, Biology, Veterinary Medicine, and Management, which was published just 2 years ago by Cambridge University Press, and really discovering some of the fundamental knowledge about giant pandas' life history and physiology. He has more than 260 publications in the peer-reviewed literature and 54 book chapters. And this work covers more than 50 vertebrate species. So he's truly been one of the pioneers in wildlife conservation and taking science and applying it toward effecting positive change for managing wildlife. And with that, I'd like to introduce our keynote speaker, Dr. David Wilt.
Well, thank you very much, Alex. I would like to begin this afternoon by first thanking Dean Kotlikov and especially Alex Travis for the very kind invitation to come back to Cornell University. It's been a long time since I've been here. John, I've noticed you've gotten a little grayer since I was here last. It's a pleasure to be here, not only to speak, but also to talk about perhaps some important partnership opportunities between our two prestigious organizations, Cornell and the Smithsonian Institution. So I'm very excited about that. And lastly, just to be here during the symposium today. I'm very impressed with the scope and the depth of what I've heard today. It's very exciting, and it's sort of reminiscent of what has happened at the Smithsonian Institution. You know, we're this huge, complex organization, and we occasionally have these calls for people to come in and talk about a particular topic like wildlife conservation, and we're really amazed that they're like people down the hall that are doing things that are really interesting and interrelate to what we're doing. So I hope that has happened today, and we look forward to working with all of you more. I was a bit challenged today in trying to focus down on what I would talk about, because we do a lot of conservation biology throughout the various museums and research centers all over the Smithsonian, and even the National Zoo's Conservation and Science Directorate is pretty complex. Most of my talk today is going to be rather philosophical, but I do want to encourage those of you, especially those that came in later today, if you have the opportunity to look at some of the posters that we brought along for the various centers as part of our Conservation and Science Directorate that are out there that talk about the mission and some of the specific kinds of things that we do. So what I thought I would do today is to tell you a little bit about our science group at the Smithsonian's National Zoo, and then to share with you a few experiences over the course of the last 30 years that have really focused my attention and the attention of my group on how what we think some of the priorities are. And you can actually see some of them listed here in terms of this whole need to develop scholarly databases for species. And for those of you that know me, you know that I've become a wildlife species advocate for the last 25 years because most of the resources that we have access to, we in the scientific community, with respect to animal research, are directed towards what I call the common species. People, cows, pigs, goats, rabbits, laboratory rats. There's nothing wrong with that, but we also need to direct research resources to these thousands and thousands of vertebrate and invertebrate species that are out there. And in doing that, what we do is we generate the basic knowledge that allows us to either deal with extremely rare species, or as we heard this afternoon in a very excellent presentation, this whole situation with overabundant wildlife as well. And we have to have that kind of data set first, and I'll actually tell you a little story about how I learned that. I really like my job, and one of the reasons I like my job is that we get to do basic research, but then we're able to convert that basic research into applied problem solving, and we can actually help species. And what I also really enjoy about this job is that even though I'm a reproductive biologist, that's not the only thing we do. I work extensively with veterinarians and behaviorists and nutritionists and geneticists and, oh yes, economists and sociologists and others to deal with this whole human dimension. So that's what's really fun, I think, about dealing with wildlife conservation, the ability to integrate all of those disciplines together to deal with holistic issues. And then the last really important opportunity here is in terms of partnerships. And I've said repeatedly that being involved in serious conservation biology is not for lone rangers. You just really can't make great progress working by yourself in the laboratory or in the field. You really need to work with others in very strong partnerships. So I will give you several examples today of how we are working in partnership. So let me begin by telling you a little bit about the Smithsonian's National Zoological Park. The National Zoo, the Nation Zoo, your zoo. It is located primarily in Rock Creek Park. There's 163 acres. That's the public facility that you probably all know about. But we also have the Conservation and Research Center, which is located about 2 hours west of Washington in the foothills of the Blue Ridge Mountains near the town of Front Royal, Virginia. This is a very, very special place. It's got a really interesting history in that it was an old Army remount station. And most of the historic barns that you see here were built in the early 1900s, and this place was used to produce thousands and thousands of horses and mules for the military. And the National Zoo acquired the property back in the mid-1970s. We've converted over most of the barn facilities for a variety of research and recovery programs for endangered species. One of the really important aspects of a place like the CRC is space. We can do things that traditional zoos cannot do. We have 3,200 acres, which is abutted right up against the Shenandoah National Park, so we're very important in terms of maintaining that green space out there because there's a lot of urban sprawl, of course, moving westward from Washington, D.C. This central core area is really made up of some of the primary captive breeding facilities. However, you can see lots of these green areas which are what best could be described as advanced secondary growth forests that are used a lot for our ecology studies. So we too study a lot of the native Virginia wildlife. We're very proud of the quality of our Conservation and Science Directorate. Arguably, we have one of the largest groups in the world. We have about 150 people, about half of those located at the Rock Creek site in Washington, D.C., the rest of us out at the Conservation and Research Center. There's 35 full-time scientists who are absolutely devoted to capacity building. We've talked about that a lot today. That's really what we all live for, is creating this next generation of conservation biologists. So I checked the other day, currently we have 15 postdoctoral fellows, and even I was surprised to find out that we had more than 70 graduate students. We are not a degree conferring institution, so all of our graduate education is done in partnership with universities. Either local universities or universities scattered throughout the United States or even abroad. The Conservation and Science Directorate is organized across these 5 centers, and that's what the 5 posters address out in the hallway. There is a Conservation and Evolutionary Genetics Center, which is led by Rob Flesher. Francisco Dahlmeyer deals with our Conservation Education and Sustainability Center. That is largely focused on our extensive training programs, and I'll share with you some of that information a little bit later in the presentation. Russ Greenberg runs our Migratory Bird Center, and John Seidensticker runs our Conservation Ecology Center, which is mostly located out at CRC and has a very extensive GIS and remote sensing component. The Center for Species Survival is the group that I have the pleasure of leading, and we are divided into 2 departments, the Department of Animal Programs, which is really focused on managing those various endangered species populations out at CRC, both mammals and birds. And that also includes producing animals for reintroduction, and I'll share some of that with you a little bit later as well. It's a bit of a unique situation because in most zoos, the animal collections are managed by curators, who normally aren't necessarily scientists. So in a reorganization that we did a couple of years ago, it was decided to put the animal collections under the science staff, which has turned out to be really good because it gives us more accessibility to the endangered species, populations for the various research projects. The other group is the Reproductive Sciences Group. This is a bunch of folks that I've had the pleasure of managing for many years. It's a group of about 30 senior scientists, postdocs, graduate students, technicians, and these folks are terrific. They are largely focused on understanding reproductive mechanisms across a diversity of species in 8 programmatic areas. People say, well, what do you work on? I generally say, well, we work on everything from fish and frogs to elephants. We are very interested, and you'll hear this throughout my presentation this afternoon, on looking at the impact of losing gene diversity in natural populations and how that influences reproductive fitness. We also do assisted breeding studies, And we're very much involved in ART technologies, including lots of work that's done with respect to non-invasive hormonal monitoring. I'm not going to talk about any of that this afternoon, but in many cases, these assisted breeding technologies are largely, again, focused on generating basic knowledge for each of these individual species, these unique species. And I'm going to talk about that a little bit as well. We largely focus on endangered species research, but I did want to mention the importance of doing laboratory animal research. And for more than 30 years, we have focused a great deal of attention on laboratory research with both the domestic cat and the domestic dog, largely for reasons that these species serve as excellent models for more than 40 human metabolic and genetic disorders. There are also a number of reproductive mechanisms that are conserved between the domestic cat and the human, and the domestic dog and the human, and that has been of interest to the National Institutes of Health, so we've generated a substantial amount of funding over the years for that purpose. And then lastly, what we learn about domestic animals does have application with respect to applying it to endangered species of, in this case, felids and canids. And I'll give you some examples with respect to that as well. I also wanted to acknowledge, in terms of all of these partnerships that we've talked about today, that Alex Travis and his group have been terrific, and they actually are partnering with us on many of our dog and cat studies. So thank you, Alex. All right, well, let's talk a little bit about the wildlife species. And I would like to say that, you know, I've always dreamed of being a wildlife biologist, and I had all this planned out when I was an undergraduate student, but that would be a lie. This all occurred totally by accident. And in the late 1970s, I was doing biomedical research, actually at the National Institutes of Health, and I met the head veterinarian there, a gentleman by the name of Dr. Mitch Bush. And Mitch came to me because he knew that I was doing research with domestic cats, and he said, you know, Dave, he said, we're having serious problems in breeding our cheetahs at the National Zoo. And we really don't know why, but he says, you do artificial insemination in cats. And I said, yeah, we do some of that. But I said, you know, I also grew up on a farm. I went to an ag school, and I've got a little experience in doing artificial insemination in cows, too. And he said, well, this is great. He says, I got this great idea. Why don't we just artificially inseminate cheetahs, and we'll get over this problem with the fact that cheetahs don't breed very well in zoos. And I thought, you know, this sounds like a great idea. And Mitch and I finally looked back at this as sort of our quick fix phase in learning about the difficulties of working with endangered species. So this is a photograph that dates back to 1980, and it shows Mitch and me in South Africa. And what we're doing here is we have a cheetah anesthetized on the table. And essentially what we're doing with this cheetah is we are collecting his semen using cattle technology. And then we use cattle technology to evaluate the sperm, to process the sperm, and even to cryopreserve the sperm. We put it in a liquid nitrogen dewar, and we put it on a plane, and we shipped it back to the United States. And then the plan was to put this sperm in cheetahs that were living in North American zoos. So to be able to do that, of course, we had to synchronize ovulation in the female cheetahs, and no one had ever done that. So we decided, well, we'll use cow hormone therapy to stimulate the cheetahs to ovulate, and then we'll just AI them like you would a cow. So I'm going to show you this vast amount of data that we generated from the study, which is actually on this one meager slide. And essentially what we did is we took 30 female cheetahs that were scattered at zoos all over North America. We traveled to those zoos after we treated the females with hormones, and we deposited the sperm. And you can see that we actually got a fair number of these animals to ovulate with the cow hormone technique, 23 of the 30. And we even got a fairly normal number of ovulations. But the bottom line was that not one cheetah became pregnant. The study in this respect was a total failure. Now you have to look at this particular set of results in the context of where I was in my career. Okay, I was a young postdoctoral fellow, really ambitious, wanted to make a name for myself. but realized that to do that I needed to publish papers. And we just spent about 2.5 years and a lot of money traveling to Africa, and this was the total set of our data. And I didn't even feel like we had enough to publish. So I was feeling really sorry for myself one night, sitting in my office thinking about, well, okay, so we used cow technology in the cheetah, and I had this epiphany, and that is that a cheetah is not a cow. And it does look ridiculous up here, but it does illustrate very well how incredibly naive we were back then to believe that basic knowledge and tools that had been developed over many, many years for one of the most common herbivores on the planet, the cow, could have any application whatsoever to one of the most specialized predators on the African plains, cheetah. It was a crazy idea. So in reality, it was like one of the best aha moments of my life because it changed the way that we did business. We went from this quick fix approach to what I call the species specificity phase, where we realized that every species, whether it was a cheetah or a Pallas's cat or a gorilla or a copy, didn't just deserve cow tools, but it deserved species-specific attention and hypothesis-based research. And in the context of the cheetah, over the course of the next 15 years, we published about 15 peer-reviewed publications. And not just in reproductive physiology and endocrinology, but also in behavior and genetics. And over time, we generated this scholarly knowledge that actually then allowed us to begin to consistently produce cheetah offspring by artificial insemination, whether it was by using fresh sperm or, in this case, this cub that was produced in 1995 from frozen sperm that was collected from a wild male in Namibia that had been captured by a farmer and was scheduled to be shot, and we got a call to go help save the cheetah. When we saved the cheetah, we also collected his sperm and we brought it back in the United States. It took us 15 years to produce this cheetah cub, but it had nothing to do really with the time. It had all to do with how much scholarly knowledge that we needed to understand basic cheetah biology. I'm always amazed at the remarkable differences amongst species. And over time, we've learned that not only is a cheetah not a cow, a cheetah is not a domestic cat is not a tiger is not a leopard. The reproductive mechanisms are as diverse, even within a taxon, as the phenotypes of the species themselves. And this is very, very important to remember, and really one of the reasons for our highest priorities. I also want to point out, and I'll be talking about this throughout the presentation today, the importance of genetics. The public, I think, when they go to the zoo and they see a small endangered species, a newborn, they think immediately that it's a valuable animal. But what we really focus on in zoos is something we call genetic management. It's not just about the production of offspring. It's about the quality of those offspring. And it's considerably different than how we think about the management of, for instance, livestock species, where we're looking at growth rate in cattle or milk production. We don't select for any of those particular characteristics when it comes to wildlife. Really what we're looking at is something we call genetic diversity. We're trying to make sure that the animals that we are managing in zoos today have the same sort of genotype as their founders, as their founder representatives, those animals that come from the wild. And we can actually work with population biologists and geneticists to try to figure out what that target should be. And in general, our population biologists tell us that what we really need to do is manage about 90% of all existing genetic diversity for the next 100 years. So we're looking over very long time spans to try to retain genetic diversity. So why do we care about that? Well, the fact is that we are scared to death of inbreeding depression. And I'm sure you've heard of stories about what happens when you inbreed livestock. And in the late 1970s, there were a couple of papers that were published that had to do with zoo populations that were just left to breed without any management and the insidious effects of inbreeding. But what I want to share with you is one of the most interesting case studies in terms of a wild population. This is actually what is occurring in many wild populations now as a result of fragmentation of habitat. And this involves the case of the Florida panther. Now, I think all of you know that there are many mountain lions out there, that they're indigenous to the western United States all the way from Canada through Mesoamerica and South America. But there's also a relic population of mountain lions or pumas existing down in South Florida. And when we got invited to participate in this project, it was in the mid-1980s. There were thought to be then about 35 Florida panthers remaining. Now, this is a really good example of getting invited by, in this case, the Fish and Wildlife Service in the state of Florida, a large group of partners. We got together, we put together a plan, developed partnerships across institutions, and a friend and colleague of mine by the name of Steve O'Brien, a Cornell graduate from the National Cancer Institute, took his team down, and they looked for genetic diversity in that Florida panther population. And the bottom line is they could find none. And in Steve's words, it was like looking at an inbred strain of laboratory mouse. Well, what we did is we took our team in to look at male reproductive fitness by collecting sperm samples from animals in the wild. And the first thing we observed was this high proportion of abnormal sperm in the semen samples of these animals. 94% of the sperm were structurally malformed. Only 6% were structurally normal. And what was particularly intriguing was this one particular defect called the acrosome, which is that membrane over the head of the sperm that plays such an important role with respect to fertilization. And so 42% of the sperm had this abnormal acrosome. And when we put these sperm on puma eggs in our in vitro fertilization, there was no interaction. So we were under the strong assumption that there was some dysfunction going on with respect to these sperm. At the same time, the team was making these other observations of an atrial septal defect in a hole in the heart of animals that happen to die, cryptorchidism, which is the defect where one or both testes are retained in the abdominal cavity, vaginal papillomas, and a very high seroprevalence to these infectious pathogens that certainly everyone in this room is familiar with. Rabies virus, distemper virus, calicivirus, and FIP. In short, the Florida panther was sort of this textbook case of what happens when you go through a bottleneck, when you lose genetic diversity. And in this case, this has been a really difficult problem to mitigate because once you go through that bottleneck, it's hard to recover. Let me give you another example. This is the black-footed ferret, Mustela nigrippus. This is a species that lives in the Great American West, the Great American Plains, all the way from southern Canada to the Texas-Mexican border, in an area which is contiguous with the prairie dog. And why is that? Well, that's because the prairie dog is really the primary prey species for the black-footed ferret. Mostly it eats prairie dogs and it lives in its burrow. But throughout the early half of the 20th century, and actually through to modern day, there were concerted efforts to poison out prairie dogs. And in the early 1900s, of course, this was because there was great interest in converting the Great American West over for agricultural lands. And so the government sponsored these very effective poisoning campaigns that resulted in the death of thousands and thousands of prairie dogs, the loss of 98% of the habitat, and thereby the demise of the black-footed ferret. And I can clearly remember as a postdoctoral fellow in the mid-1970s and sort of towards the later end of the 1970s, hearing about the fact that no one was able to find any black-footed ferrets. Now, there were none in a zoo. No black-footed ferrets. They were all in the wild. The bottom line was that in the late 1970s, the Fish and Wildlife Service actually declared the species extinct. Now that's hard to believe, having now actually worked with this species over the course of a number of years. This is a very charismatic mammal that we would actually have let go to extinction. Fortunately, there was a dog who was very famous by the name of Shep up in Wyoming, and one day he killed a black-footed ferret. He found a population, he killed the ferret, brought it to his owner back at the ranch, laid it on the porch. The rancher was pretty savvy. He knew what it was. He called the state of Wyoming Fish and Wildlife Service authorities. They rushed out and they found this small population of black-footed ferrets. Now, rather than rescue it or divide it, they decided, and it was an ill-conceived idea, to leave those black-footed ferrets in place. And they were left there for the course of the next couple of years, where the population gradually grew to 129 animals, after which it was affected by a disease epidemic, and it crashed. And the population declined to a total of 18 animals. They eventually did find 18 animals. I can clearly remember in 1986 getting a call from a colleague of mine saying, "Hey, Dave, could your group get involved? We've found a total of 6 black-footed ferrets. We're wanting to know if we could use embryo transfer to help save the black-footed ferret." Well, fortunately, they found a total of 18 animals. Those animals were brought into a captive breeding facility up in Wyoming, where they produced kits for a couple of years. And then in 1988, our Conservation and Research Center was the first organization outside of Wyoming to get black-footed ferrets. And we received them in part because we'd had a lot of experience breeding small mammals, and we also, again, we have all this space, so we could create various types of artificial habitats. We also actually imported some prairie dogs, and we had them build their own little prairie dog town so the black-footed ferrets could live in the burrows that the prairie dogs built. And we also started a very intensive research program. And because there were only 18 founders, it was really very important that every animal have the opportunity to reproduce. And animals, of course, are like people in that they have sexual partner preferences. This was certainly the case with respect to black-footed ferrets, and some of those animals failed to reproduce. So we were asked to develop artificial insemination technologies. That actually took a couple of years and lots of basic research, but I want to just show you in this summary slide sort of the chronology of events. So you go back to 1986, where we have the near extinction event, where we have the recovery of 18 animals, and then lots of intensive research from lots of different angles, lots of different disciplines. Not just our zoo, but other zoos were involved in this, and working together in partnership with state and federal authorities, and lots of natural breeding has occurred. Now, out at the CRC over the course of the last 20 years, we produced over 500 black-footed ferrets, including 135 black-footed ferrets by artificial insemination. And this is work that's primarily led by Dr. Joe Gale Howard from my laboratory. You're going to hear Joe Gale's name a couple of times over the course of the next few minutes. But Joe Gale has done amazing things in terms of developing AI. And here's the really, I think, important number, is that all of these institutions together have produced over 6,000 black-footed ferrets from those original 18 founders. And this has allowed reintroduction back into the American West, including now the existence of over 750 black-footed ferrets in the wild. So you look at a slide like this and you think, okay, this is how science has helped save an endangered species and recover an endangered species. However, remember again that we were dealing with a group of only 18 founders. A bottleneck occurred, and it's really quite interesting that over the course of the last few years, we've started to see some abnormalities pop up in the population, both in captivity as well as in the wild. And this includes increased numbers of abnormal sperm, some uterine or renal aplasia. Well, you can read all these. Cryptorchidism, again, the retained testis defect, and overall smaller body size. So again, once the population goes through a bottleneck and you lose that heterozygosity, you're going to have some complications. And, you know, God is not making any more black-footed ferrets, so you have to end up working with what you have. Now, there was some encouraging work done this year by Joe Gale, and I think goes back to the importance of banking down biological materials from animals over time, and that was brought up a couple of times during the day. Even though it's very, very difficult to collect and freeze semen from black-footed ferrets, we have been able to bank down a fair number of samples, and this year Joe Gale actually produced 2 male kits from artificial insemination with frozen sperm, where the males had been dead for 9 and 10 years respectively. So this is not quite like going out and finding that new population of black-footed ferret, but it is one way to mitigate against some of this genetic drift that does occur naturally over time. So it's very important to bank down biologicals. The last example I want to share with you over the course of the next few minutes has to do with the giant panda. We have spent a lot of our careers, a lot of time during our careers, working on these species that have gone through bottlenecks. And so I've been an advocate for what I like to call preemptive strikes. Why don't we spend more time working on species that we believe that are endangered but still have plenty of genetic diversity, and see if we can turn around reproduction and management in those populations now. And I think the giant panda is really a good example, because we know there's a lot of gene diversity in giant pandas. Has anybody been to the National Zoo to see the giant pandas? Okay, good number. This is Mei Xiang and Tintin. They're ambassadors from China. They're on loan to the people of the United States, and they're maintained at the National Zoo. There are 3 other zoos in North America that maintain giant pandas: San Diego, Memphis, and Zoo Atlanta. What fascinates me about giant pandas is their reproductive biology. The female is only in estrus and sexually receptive for 24 to 72 hours annually. She's monestrous. She comes in at heat once for a very short period of time. That's only 1% of her entire annual lifespan is devoted to reproduction. So if you happen to be the reproductive biologist at the National Zoo, then you got a lot of pressure putting these animals together and getting a pregnancy. And I don't know how many of you have ever seen the mating posture in a giant panda, but these are photographs provided to me by a colleague of mine by the name of Ron Swaisgood at the San Diego Zoo, who studied the breeding behavior of giant pandas up at the famous Wulong breeding reserve up in the Wulong National Nature Reserve in Sichuan Province. And they have these 2 copulatory stances, one with the male standing up and one with the male sort of sitting down. And this is an ideal kind of situation. Now I want to show you the breeding posture for Mei Xiang and Tintin. This is what we've been going through for the last 4 or 5 years. And Mei Xiang, when she comes into estrus, for whatever reason, she sort of lays down and her back legs sort of splay out. And the male giant panda during estrus gets very, very aggressive. He's basically sort of going through a rut where his testosterone levels are very high. And so he's extremely frustrated, as you might imagine. He grabs the female by the neck. He grabs Mei Xiang by the neck, and then he sort of ends up trying to get behind her, and in the end makes it all worse because he ends up standing on her back. And so they have never successfully copulated, which is one of the reasons, I guess, we have a reproductive sciences department at the National Zoo, because then we can bring in the team and do artificial insemination. So this is Joe Gale doing an artificial insemination. And this was actually the one from 2005. This is an endoscopic AI with fresh semen that resulted in the birth of Taishan. And Taishan arguably is the most popular life form in Washington, D.C., which these days isn't necessarily saying too much. But his birth in 2005 was seen as a milestone at the National Zoo, and people got very excited about this, and they look at Taishan and they say, oh, this is how the National Zoo is contributing to conservation of this endangered species. I don't like to dampen their enthusiasm, but the reason Taishan is exciting to me is because he gets people to open their wallets and give money for our program to support conservation activities in China. And the reason you don't see many zoos holding giant pandas is because it's incredibly expensive to do this. You have to actually show the Fish and Wildlife Service that you are doing in situ enhancement in China. And to do that, you have to invest a lot of money in China. And so every year we spend between $1.2 and $1.4 million annually, either providing it to the Chinese to affect their own conservation activities, and or actually focusing on two areas that have to do with lots of capacity building in the context of saving giant pandas and their habitat in the wild, or focused on trying to turn around the captive breeding program with respect to those ex situ giant pandas. So I want to tell you a little bit about that. The question always arises, how many giant pandas are out there? How many giant pandas are enough? And if you read an article in the newspaper about giant pandas, I can almost guarantee you they will say that there are 1,600 giant pandas in the wild. And that's because that number comes out of a national survey. But the fact is, nobody knows how many giant pandas are out there. I don't know. The Chinese don't know. World Wildlife Fund has no clue. And that's because this habitat is incredibly rugged. These are very steep mountainous bamboo forests. They're almost like rainforest. And it's very hard to even walk around in those forests, let alone to count giant pandas. So nobody really knows. And as I tell people, it's less about the total number of giant pandas than it is about the quality of the habitat. And here's the important point from this slide, that there's lots of fragmentation. I'll show you a map a little bit later. Lots of fragmentation in the wild. More than 55 isolates comprised of giant pandas. And some of those isolates contain only one or two giant pandas, purportedly. So those are hardly self-sustaining numbers, and there's no corridors of genetic exchange between those isolates. And as a result, that's why the species is highly endangered. There's another population of giant pandas that live ex situ in captivity. And we know exactly how many giant pandas live in captivity. At the end of last year, in the 2007 stud book, there were 241 giant pandas. Now these animals are incredibly important because they are ambassadors for the species and for China, but they're also a research resource. I mean, if we have graduate students who are interested in studying sperm metabolism in giant pandas, the only way they can do that kind of research is work on captive animals. And so we do a lot of those kinds of studies in China, primarily at the two main breeding centers. One is at the Wulong Breeding Center, which was the one that was recently devastated by the earthquake, and the other at the Chengdu Research Base of Giant Panda Breeding, which is on the outskirts of Chengdu City. That's where the majority of giant pandas are within China, and then there's a few outside of the country, including in the United States, as I described a few minutes ago. Now, I haven't had the opportunity to, we talked about partners a while ago, I haven't had the opportunity to introduce my life partner, my wife, Susie Ellis, who, for those of you that weren't here a while ago, we were talking about the International Rhino Foundation and the partnership the International Rhino Foundation has with Cornell University. Well, Susie also was involved with us with respect to this giant panda study. And in 1996, we had the opportunity to go to China at the request of the Chinese government to talk about the captive population of giant pandas. And we spent a week listening to what the Chinese had to say. And they were essentially concerned that the population was not self-sustaining, that more giant pandas were dying every year than were being born. And we heard a lot about health problems and lack of reproduction. And at the end of that week, we decided that the only way that we could really get a handle on this was to do what we called a biomedical survey. And so Susie and I were privileged to lead a team of Westerners over to China, not as missionaries, but to work hand in hand with our Chinese colleagues to get our hands on as many giant pandas as possible. And in 1998, when we began the actual biomedical survey, there were 104 giant pandas in captivity in China. And we were able to get our hands on 61 of those we took along these various specialists. And this is an example of how you integrate all these scientific disciplines together to try to figure out what's going on with a population. And we did. We learned a lot, so much so that we were able to produce this book on giant panda biology. But let me just illustrate some of the factors that we discovered that were limiting reproductive success in giant pandas. Aggression. I've already talked at length about the short window of estrus in the female. Well, in many cases, you introduce the male and female together, and the male would rather fight with the female than to mate with her. So that's another huge problem. I suspect that everybody in this room knows that giant pandas are these big old bears that live on grass. They eat bamboo. And what the Chinese have encountered is that when they manage 20 or 30 giant pandas in one place, it takes a lot of bamboo. And so what they've tried to do is create these artificial diets. And this is one of these diets, which is a high-protein bread meal. And what they do is they feed it warm to giant pandas. So imagine if you were a giant panda and they gave you a stalk of bamboo or some nice, warm, palatable, high-protein, what would you eat? Well, they eat all this stuff. And the more of this stuff you eat, the less likely you are to be healthy or to reproduce. And we also encountered something that we classified as the stunted development syndrome, which we actually saw in 13% of the population. These are giant pandas that are about 2/3 the size of a normal giant panda. They have lots of health problems. Their abdominal cavity is filled with ascites. They've worn down all their teeth, and not one of these animals has ever reproduced. We also encountered some interesting health cases, some of them very simplistic. This is a female that the Chinese simply call number 20. She doesn't even have a name. She was a founder animal. She was rescued from the wild. So genetically, she was really important. Here you see she's sitting here partially sedated with a dart in her leg. And they told us that every year they bred 20. She copulates, but she never ever produced any offspring. And when we did the biomedical evaluation, it was discovered that she had a simple cervical vaginal infection that was cleared up immediately with long-acting antibiotics, and this animal has reproduced repeatedly since that biomedical survey. This is a more serious situation. This was another founder, squamous cell carcinoma. By the time we got to this animal, including the people up at Wulong, he was near death. But because we knew so much about the reproductive physiology of the male, it was possible to collect semen from this animal. And one of our students, Dr. Wan Yan, was able to use artificial insemination to deposit sperm in a 5-year-old female, and that's a pretty young female, and she produced this cub. So despite the fact that this male was euthanized, and he died, obviously, his genes live on in the captive population. Lots of reproductive biology studies, and we've talked a little bit about sperm cryobanking combined with artificial insemination. In many cases, we run into species where the sperm are resistant to low temperature. In the case of giant panda sperm, here's a species where the sperm are fairly tolerant to cold temperature. So it's relatively easy to cryopreserve giant panda sperm. And as a result, the Chinese have developed a rather extensive sperm bank, and they consistently use artificial insemination. They even artificially inseminate giant pandas that naturally breed. So this has become a part of their genetic management program. So there's been a lot of research, not just in reproductive biology, but all those other disciplines. And over time, there's been some positive results. And one of those is that you can now go up to these breeding centers during this time of the year, this is really the birthing season, and you can begin to photograph piles of baby giant pandas. This started about 6 or 7 years ago. And I showed this the other night at a presentation, and somebody said, well, those are beanie babies. And I said, no, that's the real deal. And the Chinese take great pleasure every year in getting these group photos together with their giant panda cubs. And every year, this number has grown. And what's particularly exciting now is that on average, they are producing about 30 surviving giant panda cubs per year. And even this year, during all the disasters associated with the earthquake, there were a total of 25 giant panda cubs produced. So they're doing great. And the natural question is, people say, well, how many giant pandas do you actually need? Well, this can be calculated. If you know how much genetic diversity, if you know how much gene diversity is in the population and how much you want to save, you can actually calculate how many animals you need to maintain in the XC2 population. And we know what this number is. John Ballou is our population biologist, works very, very closely with the Chinese to calculate this number and to make sure that we create these genetic matches. The number now, remember the number in 1998 was 104. Well, at the end of last year, we were at 241. So the population has doubled in the last 10 years, and then they've produced another 25 cubs this year. So within the next year, and certainly within the next 2 years, they will have reached that targeted population of giant pandas. And that will mean that they then have a self-sustaining captive population, an insurance population. And then if and when they decide that they want to do reintroduction, they will have this source population that they can harvest animals from for the reintroduction programs. But let me emphasize that all of that work involved with the managed care populations in captivity really means nothing unless you're able to maintain giant pandas in nature. And I just want to spend a couple of minutes quickly sharing with you some of the work that we do in the field. This is a trip camera photo of a giant panda that's walking along the trail. I'll talk a little bit about mammal surveys in just a second. But this is wonderful habitat, and it's really a high priority to figure out how we can maintain it and how we can expand it. And we think the key to that, the key to everything we do with respect to species conservation biology, is capacity building and training. And so we've been invited by the Chinese to do a lot of training overseas. And I just want to give credit to a couple of people. Melissa Songer is one of our field ecologists, and she works in our GIS and remote sensing laboratory. And her goal really is to determine how much giant panda habitat is still out there, and then to create the habitat maps, because amazingly we don't have maps for all of these more than 60 reserves now. So she is doing that, and she's doing that under the umbrella of training. So we do these courses every year. And also, we read a lot, we hear a lot about all this money the Chinese have, and yet that money is not really trickling down to the reserves, so they really don't have a lot of the equipment that they need to do the conservation management. So we're trying to bridge that technology gap by providing computers and GPS and satellite imagery and software and taking these young professionals out in the field where they actually get some field experience and ground truth the quality of those reserves. This is a relatively new and I think pretty exciting map created by Mel's students over in China. This is the entire giant panda range that involves three different provinces. And the light blue areas that you see, these are the managed areas. These are the official, the national or provincial reserves. And what I want to point out to you is that all of these red areas, particularly here in the north and here in the south, that is suitable giant panda habitat that's been found. And so what we do is we share that information with the Chinese government, and they have really pledged to restore as much of that. And in doing that, we'll hopefully create these corridors which will allow the movement of giant pandas throughout the range, or at least through most of the range. And Bill McShay is our field ecologist that is working with our Chinese colleagues with respect to doing mammal surveys. And so sometimes the photographs, for those of you that are familiar with this technology are not perfect, but you can get an idea of what's out there. And I think what's most exciting to us and what we get a lot of fun out of is seeing how excited our Chinese colleagues get by discovering these other species that are living in these reserves. Anybody know what this is? Shout it out. Takin, very good. This is a species that you don't see a lot of in zoos. It's very difficult to actually keep in zoos, but it's sort of a cross between a cow and a goat and a moose and a bear and wildebeest. But as a result of this study, we've actually started some studies on migration of takin, and we have radio collars on 7 takin over there. And seriously, I just got this photograph this week. This is up in the Shanxi province, up in the north. Our first photograph of a leopard, a large leopard, who actually happens to be sitting in a stand of bamboo, which is, of course, panda habitat. So that's always exciting to a cat person like myself. Lastly, I think probably more than the science that we've done or participated in China, if we're going to leave a legacy, I think it is with this training. We're very proud of the fact that more than 700 Chinese professionals have gone through these various training courses that we have conducted there. And we started out in 2000 with respect to our training programs, and we sort of joked around and said that our goal is to train ourselves right out of a job. Guess what? We're very close to being able to do this. Almost all of this work now is being done solely by our Chinese colleagues, and I think we'll be probably out of China, at least out of these projects, by the end of 2010. Lastly, I just wanted to show you this photograph. I don't know whether any of you have had the chance to go to China and go to a scientific meeting in China, but there's this tradition, and Susie and I always get a kick out of this, because you go and there's this elaborate opening ceremony with flowers, and the mayor comes in and they do all of these introductory speeches. And then immediately after this exercise, everybody goes outside to get a group photo. And it doesn't make any difference whether it's 95 degrees outside or 10 below. You go outside and everybody waits to get this photograph taken. And I show you this photograph because they all always look the same. Everybody looks sort of dour, but the important part of the photo is that it is consisting of such diverse people. You have people from competing federal ministries in Beijing, competing provincial ministries. You have competing breeding centers, zoos, universities. And why this is so amazing is that 10 years ago, these folks did not have the opportunity to even attend a meeting like this, let alone to sit down around a table with laptop computers and solve problems. So I think this is the reason that so much success is going on now with respect to giant panda conservation in China, and why it's extending to other species as well. And I think this is really a good example for the rest of us, because the conservation programs that we've been involved in, the successful ones are the ones that involve this kind of diverse partnership. It's really related to that whole human dimension that we were talking about throughout the day, and how important this is. And speaking of the human dimension, this is the human dimension group that I have the privilege of leading. And I just wanted to acknowledge again Joe Gale Howard, also Janine Brown, who runs our endocrine lab, Steve Momford, who is now the head of our Conservation Science Directorate, and Budhan Pukarenthi. And the 5 of us have been together now for more than 100 years, which means, I guess, that we like each other and we like our job. But most of all, we greatly respect this very talented group of young people who are so dedicated. And these are the folks that are out doing the work, taking the risk, and creating those stories that allow me to come and to share them with you. So I want to thank you very much.
read that a bamboo magazine dies after 100 years. So you have to have different areas.
Actually, the reason that has been a serious problem in some reserves, but the fact is that in most reserves there's several different types of bamboo. So one will die off and the pandas will switch to another. But there are certain reserves where it's really been a problem, and there have been significant amounts of mortality associated with that. And this really has a lot to do with the reforestation efforts that the Chinese are also planning, so they're beginning to address those issues as well. Other questions? Yes?
So this is sort of a philosophical question, I guess, but what are your thoughts on, I guess, the evolutionary implications of using all this AI technology? Do you think that's sort of a new form of evolution, or?
Well, no, I don't. In large part because what we're doing is we're using artificial insemination to match up genetically valuable individuals. But we have lots of examples, for instance, of some of these cheetahs that have been produced by AI, that when they mature, they naturally breed. So there's no evidence that we are losing some sort of mating fitness gene as a result of doing the artificial insemination, so we really don't worry too much about it. Your question, I think, is relevant to being able to make another point, and that is, we do embryo work in our laboratory, but it is more along the lines of using it to understand the whole process of sperm-egg interaction and embryogenesis. I don't know of anyone who's actually using embryo transfer to genetically manage an endangered species population, and there's lots of reasons for that. But we do feel that artificial insemination is probably about the only technology that we really need with respect to genetic management of endangered species. It works. It satisfies our needs. Yes?
In regard to that, can you induce estrus in the panda?
No. No one has successfully induced estrus in the panda, and it's really quite interesting because being such a political animal, it's really difficult to go beyond doing just artificial insemination. The Chinese actually have discussed the opportunities and to do some ovulation induction, and I will tell you that about 10 years ago, they did share some data with us where they gave massive doses of serial injections of equine chorionic gonadotropin to 3 giant pandas and caused some enormous problems with respect to those animals. So it was unfortunate because it might have worked if they had designed the experiment appropriately, and so that's really left a bad taste in our mouth. So we haven't really pushed that at all. Yes.
Outside of the scientific community in China, how much support do you have for giant panda and land conservation for the panda within those areas? And do you get out and have the opportunity to talk to the local people about it?
Yeah, well, your second question first. With respect to a lot of the mammal survey work, it's been closely tied to local communities. And in fact, it's really, we're very interested in Asiatic black bears because of black bears that are not really seen as a high priority species for conservation. In fact, are captured and used in biofarms. And so we felt it was really necessary to get out into local communities and get a handle on what they thought about wildlife. And so that's a whole area that needs a great deal of attention. Also in that country, there has not been a lot of emphasis on habitat management. But that also is changing, and it's changing really as a result of these training programs where we actually talk a lot about landscape management issues, and then we actually create these maps, and then the decision makers in Beijing, who frequently do not have lots of conservation background, they began to actually see these maps. And when you can see hard data, then I think they're going to listen to the fact that there is a need for some serious landscape management planning in China. But I think we're getting there. Yes, Susan.
So in anticipation of being able to release some pandas back into the wild in a
interest, you may have read in the newspaper that the folks up at Wulong did do a release of a giant panda a couple of years ago, which turned into some disastrous publicity for them because they turned it into an area where there were other male giant pandas. So that, over the course of about a year or so, that animal was harassed by other pandas and was eventually killed. So again, very good lesson learned. What we have emphasized, again, we've started to talk a lot about what the requirements are, the prerequisites before reintroduction, including knowing what's out there, both in terms of food resources as well as other giant pandas. And when I showed that map, that whole southern area down there is really a rich area for reintroduction. We think the density of giant pandas is very low in that area. And so what is being encouraged right now is to use that southern region of the Sichuan province as the test region now for reintroduction. Very few giant pandas there, so that won't be a problem, but good stands of bamboo. Yes? Tiger farm, also tiger farms in China. It's a very, very big deal, hot button issue, and it's interesting because we have very close friends who work in the Chinese government associated with CITES, and they really don't understand our opposition to either biofarming, for bear farming or tiger farming. It's, this is a culture that goes back 6,000 years, and so we have to respect that and have open dialogue, but I can tell you it's a very difficult issue. Okay, thank you, Alex.
Well, I'd like to thank David for a truly a wonderful talk, and really just very inspiring work over a course of decades that's really pushed conservation forward. No? So we were going to have a few closing remarks by Dean Michael Kotlikoff. President Skorton has a trustees meeting today, and there's lots going on, and I know the dean's schedule was changing even up to yesterday afternoon, when that got moved to a closing remarks. And here he is, just in time. So I'll now turn things over to the Austin Hui Dean of the College of Veterinary Medicine, Michael Kotlikoff.
Thanks very much, Alex. Hi, sorry I'm just a touch late, and I apologize that I haven't been able to participate more. This is Alumni Council weekend, and we've been running back from president's statements to Board of Trustees meetings, et cetera, et cetera. So but I did not want to miss this opportunity to express my appreciation for everyone that's come here and my commitment to the area of the general area of expanding our approach to the concept of the health of our wildlife species and our environment, and as well as our domestic animals and pet animals. So I know this has been a very broad conference, and I appreciate everyone that has come, and I hope this is the beginning of more interactions and more focus on this area at Cornell, and a more effective organization of our activities that we're already doing across the campus and with our friends and partners. So thank you very much for coming, and look forward to talking with you a little bit after the conference. Thanks.
As well, and all the speakers for really a really wonderful day. Again, please, if you just came in, sign up in the back, and when we get the website online, We'll have email lists for funding opportunities, special seminars, and relevant activities. Thank you.